Welcome. Thank you for taking the time to view this presentation on the guidelines for filling out your Barcode of Wildlife Project Field Data Spreadsheet. The aim of this video is to introduce you to the Barcode of Wildlife Field Information Management System, otherwise referred to as the FIMS. In this video, we will be covering the topics outlined above, specifically, what is the BWP FIMS, populating the BWP FIMS spreadsheet, required fields for standards, input formats along with importing data from existing collections, handling additional data, for example digital images and video, and an introduction to frozen tissue vials, 1D and 2D barcodes. To begin, let's briefly describe what the BWP Field Information Management System is and the advantages to using it. The BWP FIMS is a database storage system or a data repository where all BWP field data is held. The advantages to using the BWP FIMS over a traditional spreadsheet are: the BWP FIMS gives you the ability to validate your field data. This means that when you are uploading your field data to the FIMS, you can confirm that not only are your entries in the correct format, but also that you have entered all of the required field data that are needed to meet the BWP barcoding standard. With regard to safety, what we mean is that when all field data have been validated and stored in the BWP database, there is little to no chance that this data will be lost or modified, a scenario that could easily take place if your data was to be stored as a hard copy in a lab or as an electronic copy on your personal laptop or desktop. Lastly, what we mean by accessibility and transparency is that because your field data is housed in BWP's data repository, it can be easily accessed and shared with project collaborators as well as project grantors. So where does the FIMS fit into the overall barcoding pipeline? Let's take a look. Here's an overview of the BWP barcoding pipeline. The starting point for all labs is choosing target species based on BWP provided guidelines. According to the guidelines, all specimens processed must either be a priority species from your country list or a close relative of a priority species from your country list, or a look-alike of a priority species from your country list. If you have questions about these guidelines, please contact your project informatic leads or lab working group members who have access to these documents. After the target species are chosen, the next step in the pipeline is to sample the specimen itself, either from fresh material or from sampling of existing collections. Once collection begins, it's imperative to start populating your BWP FIMS spreadsheet with the sample's associated field data. With a completed spreadsheet, the next step is validating and uploading your spreadsheet to the FIMS database. Data must be validated before it can be uploaded into the FIMS database. Once the FIMS spreadsheet has been uploaded, the physical processing of specimens in the lab could begin. At this point, you will also start to populate the Laboratory Information Management System or electronic notebook with your lab protocols and experiments. The next video that we will be posting will introduce you to the Laboratory Information Management System or LIMS. Conveniently, the FIMS and LIMS databases are linked and you will be able to view your specimen's field data in the LIMS repository. Additionally, all of your field data will accompany your specimen sequence information when your records are eventually uploaded and published in GenBank. In this tutorial, we will be concentrating on how best to populate your FIMS spreadsheet with the required BWP information. Before we walk through the actual spreadsheet, let's discuss a couple of points. As highlighted here, the most important take-home message of this video is that the quality of your barcode sequence record is only as good as the field data that you have collected to support it, meaning that without exceptional field records, your sequence data will be unusable as a BWP Silver Standard barcode record. And remember, Silver is the lowest standard that the BWP will accept. If you are unfamiliar with these BWP barcode standards or need a refresher, please refer to the consultation video. The second point is that the key to successfully creating high-quality barcode libraries depend on two things. One, capturing high-quality relevant data either in the field or when subsampling repository specimens, and 2, the correct storage of the material in a way that is traceable, permits little or no user error, and allows high-quality data to be reproduced if and when necessary. With regard to this last statement, we will now briefly introduce you to the FluidX system that you will be using throughout this project. What you see here are 2D bar-coded cryovials, which will be used for both long-term storage of tissue and long-term storage of DNA, along with 2D bar-coded racks, which will hold the 2D vials, and a barcode scanner, which will read the 2D barcodes on the cryovials. The barcodes on the cryovials and plates will serve as unique identifiers of the tissue, DNA, and set of samples contained in the plate. We will be discussing this equipment later in Module 2. Here is a snapshot of the BWP spreadsheet that you will be required to fill out for this project. You will be able to download this spreadsheet from the Open Access BWP Bookshelf folder that you have access to through the BWP Google Drive. This spreadsheet contains 65 columns with terms that head each column. The terms are broken into 3 different categories color-coded as blue, orange, and white. The terms highlighted in blue represent data that must be present for your spreadsheet to be successfully validated and subsequently uploaded into the FIMS database. The terms highlighted in orange, if left blank, will not prevent you from uploading your spreadsheet into the FIMS database. However, if not eventually populated, will prevent the acceptance of your sequence into the BWP project. The blue and orange terms identified here represent specifically the terms chosen by the BWP Specimen Working Group as the most valuable metadata to accompany your barcode sequence in GenBank. Thus, you will not be able to acquire the BWP barcode flag or submit your sequence to the BWP project without all of this required data. A couple of things to point out here. There will be instances in which the required metadata will only become available after the appropriate method of acquisition has been taken, for example, final species identification by field experts. For in-country trainings, you are expected to come to the lab training with all of the required fields on the BWP spreadsheet populated, minus a few fields that we'll discuss in a moment. We've created a reference resource for you, accessible on the BWP landing page that explains each of the terms used in this spreadsheet. Now we would like to familiarize you with the BWP Terms List. The BWP terms that you see here are closely related to the terms or fields taken from the Darwin Core. The Darwin Core, as taken from their webpage, is a body of standards including a glossary of terms intended to facilitate the sharing of information about biological diversity by providing reference definitions, examples, and commentaries. This BWP Terms List is an interactive page that will allow you to access definitions of BWP spreadsheet terms and input examples, which will make populating your spreadsheet clear and easy. You can click on any one of these terms to obtain its BWP definition. Let's take a look at a few examples. The first term listed on the BWP spreadsheet is Processing Lab. According to the BWP definition, this is the lab where specimens will be processed from extraction through to PCR purification. Input examples for this field are acronyms of the institution or lab. For example, NMK-ZO, ECPay, or SANBI. When your spreadsheet goes through the validation step on its way to the FIMS, this term will be checked to make sure it contains one of the accepted BWP values. This is one example of the validation rules that we have constructed as part of our data quality assurance. If you are constructing your FIMS spreadsheet with data taken from repository specimens, please do your best to match your repository's terms with these BWP definitions and then fill the sheet accordingly. The second term listed is extraction plate. Again, according to the BWP Terms List, this is the barcode number on the FluidX plate which will store your DNA extraction tubes. For purposes of the in-country training, this column will not be populated until after you have received your FluidX plates and tubes and the DNA extraction process is complete. The third term listed is extraction well. The BWP definition of extraction well is the position of your FluidX barcoded extraction tube contained in your FluidX barcoded extraction plate, which will store your DNA extractions. Again, For purposes of the in-country training, this field will not be populated until after you have received your Fluidex plates and tubes and the extraction process has been completed. The fourth term listed is Occurrence ID. The BWP definition of Occurrence ID is the unique ID given to the specimen the tissue was taken from. It is constructed from the institution code, the collection code if one exists, and the catalog number, joined by a colon. As seen here, the BWP Terms List will provide examples of the data that the project is trying to capture through this spreadsheet. In this instance, the example used is USNM: mammals: 1234. If you are sampling specimens that have data in the form of media, such as a video or a photograph, you will be required to upload the media to either Flickr for photos or to the BWP YouTube channel for videos. These links are provided under the Associated Media in the Barcode of Wildlife Terms page. Once uploaded, these media files will be linked to your FIMS spreadsheet. Now that we have walked you through a few examples, please take some time to further familiarize yourself with the BWP Terms page. We want to reiterate that all blue columns must be completed to successfully gain access to the FIMS database and subsequently to move through the BWP barcoding pipeline. Additionally, all orange fields must be populated before these records will be labeled as barcode platinum, gold, or silver in GenBank. Once you have populated the fields required on the spreadsheet, you will be ready for the next step in the Informatics pipeline, validating and uploading your spreadsheet to the FIMS database. This will be explained to you in the next Informatics video. This brings us to the end of our FIMS spreadsheet instructional video. We hope that this tutorial has been clear and informative. If you have any further questions with regard to populating your spreadsheet, please don't hesitate to contact the BWP Informatics trainers. Their contact information is listed here. And remember, the quality of your barcode record is only as good as the field data that you have collected to support your specimen. Thanks again for watching.